everyone, and welcome to another webinar hosted by the CMT Research Foundation. I'm Susan Rudiger, the co-founder and the Chief Mission Officer at the CMT Research Foundation. But I'm also a person living with CMT. I have CMT1A, as do many people in my immediate and extended family. In fact, we can trace CMT back to my great-great-grandfather through oral history and family photos. About the time he was working on the railroad in the Canadian Plains, Dr. Charcot was characterizing CMT in Paris. There you can see my family at a family reunion, and everyone with that yellow arrow, we all have CMT. So 140 years ago, CMT was named, and now we have promising clinical trials. Thanks to the advancements in biotechnology like gene identification and the ability to make preclinical models that have CMT, the approval of gene therapies for other diseases, and of course the promising research that has been funded by our donor community, the future of treatments and cures for CMT is bright. Since the founding of the Research Foundation in 2018, we went from one active trial as a community to several now. So several trials. This is such an exciting time for the CMT patient community as real treatments are under development and being tested in people with CMT now. We've heard early feedback from people in the trials that they feel like life is 20% easier. As a woman who struggles to tie her shoes or chase my dog or find the energy to make dinner after an arduous day, 20% easier just sounds incredible. And when I think about the ability to end a family legacy, one that has lasted in my family for 140 years at least, and across 6 generations, I really do get chills. I mean, we're getting that close. We'll talk a little bit more about the current trials that are happening and what we know about the progress they're making. We'll also discuss the planned trials for CMT and give you more details about how you can find trials and how you can enroll. This is why we're here tonight. We're here to learn about the clinical trial process and the trials that are relevant to you. While we are hopeful that these trials will result in improved therapies, we know that one approval will not end CMT for everyone. So at the Research Foundation, we remain focused on our core mission statement. We fund drug development research that can lead to treatments and cures for all types of CMT. To date, our donor funding has funded 35 different projects. Excitingly, we have funded a few of those higher risk drug development projects that have led to follow-on investments from venture capital groups and pharma to the tune of like $1.2 billion. And that funding has initiated 4 clinical trials. That's right, a $127,000 investment in a small biotech called DTX Pharma, a company, by the way, who had never heard of CMT before we met them. That led to a $1 billion purchase by Novartis, and now they're in clinical trials in Canada for CMT1A. A $683,000 investment in Augustine led to an $85 million raise from private equity and pharmaceutical companies like Novo Nordisk and Lilly. They're now in phase 1 human safety trials. And an $800,000 investment in Elpida led to an $8 million grant from the California Institute of Regenerative Medicine, and they will start a CMT4J trial in March or April of this year. That's a gene therapy. So these are examples of your philanthropic dollars at work. The CMT Research Foundation was founded by patients, is led by patients, and is funded by patients and the people who love them. Now, speaking of patient leaders, I'd like to call out Laura McNeil. We're not going to put her on camera, but she's the CEO of the CMT Research Foundation. She's here tonight. She joined us at the Research Foundation in 2024. She previously was the CEO of another nonprofit focused on drug development, but this one was for Lyme disease. But Laura came to the CMT Research Foundation because she's one of us. She and her family have CMT. So our focus remains strong. And while we're encouraged by the trials planned and underway, we all know that trials are not approved treatments, and not everybody's willing to be in a trial. And it's true that trials for one type can lead to trials for many other types, but not all the trials are for everyone with CMT. So there are some therapies for CMT that are under development that can address the most common challenge. helping the nerves and the muscles talk to each other. All of us with CMT, we experience that challenge. But there are also some that can address the root cause of each type, the genetic variant. What you all might have heard is gene therapies or genetic therapies. Recently, the CMT Research Foundation welcomed Dr. Megan Drummond to the team as our Vice President of Research and Drug Development. She's a genetic neuroscientist who comes to us from Regeneron, a pharmaceutical company, where she designed, built, and dosed a gene therapy for a rare form of genetic hearing loss. 3 years ago, kids who couldn't hear received her drug, and now they're hearing everything. She essentially cured their genetic disease. Lucky for us, she's now focused on CMT. So in the 6 months that she's been here, she's had a chance to get to know the CMT ecosystem and to learn the progress that's been made and to identify those opportunities to remove the remaining barriers between early drug discovery and clinical trials. I'd like to introduce you all to Dr. Drummond. And Meg, will you share some of your findings?
Yes, I'd be happy to. And thank you, Susan, for the kind introduction. So some of the things that have really stuck out to me as after joining the Research Foundation is that, in particular, the Research Foundation does a wonderful job of supporting programs with a high translational potential. We do fund discovery science, which our portfolio includes that, but it also includes programs across the entire drug development spectrum, from early discovery work to programs that support clinical trial readiness. I think that this makes the future of clinical trials feel very tangible. What I find most exciting about the portfolio and the CMT field generally is that many modalities are in play, where the field collectively is looking at small molecules, genetic medicines, gene therapies. It's a robust and active discovery pipeline that's feeding the translational pipeline that will ultimately bring therapies to patients via clinical trials. It's leaving no rock uncovered and looking for the best solution that we can possibly find. All of this also leads to the feasibility of getting these into patients, doing so safely and in a way that can make a real difference in a patient life. Which leads me to our next speaker and why we're all here tonight. Jenneen DeFiore is the VP of Neuroscience at PPD, part of Thermo Fisher. There, she supports pharmaceutical companies design and implement clinical trials for neurological diseases. But she's also one of us in the CMT community. Jenneen's family is affected by CMT, and she lives with this experience every day. Jenneen is here to educate us about how clinical trials are designed and what that experience is like for people who participate. After Jenneen explains the details of clinical trials, I'll come back to focus on the trials that are happening right now for CMT. And then after that segment, Susan will lead a question and answer session. So please ask any questions through the Q&A button on your screen. And without ado, let's hear from Jenneen.
So hello, everybody. Thank you so much for having me. I'm just waiting for my video to come up. All right, there we go. So thank you all for coming today. I'm so excited to go through this with you. My name is Jenneen DeFiore. I'm a Vice President of Neuroscience at Thermo Fisher Scientific. And first things first, I want to thank the CMT Research Foundation for allowing me this opportunity to speak with you today. So today I'm going to talk to you about clinical trials, how they work, what to expect, and what they realistically mean for patients and families. So I want to start first with why this topic is so personal for me. So CMT isn't just something that I work on. Yes, I'm in neuroscience. I touch a lot of therapeutic areas within the neuroscience world, but it's something that affects my family across multiple generations, including these amazing people that you see on your screen. So my husband, Gregory, my daughter, Lily, my niece, Allie, my father-in-law, Gino, and my sister-in-law, who's not on the screen, all have CMT1A. I think about them every single time that I step into my office, because CMT is never abstract for me. I've seen how it affects their daily lives, their independence, and the decisions that we make as a family over time. I've also spent 25 years working in clinical research. including leading and supporting trials in rare and pediatric diseases. My focus has always been on helping design studies that are safe and meaningful for patients and responsibly moving ideas into real treatments. My family's experience gives this work its context, and it reminds me every day that behind every study and every protocol are real people, real families, just like us. That perspective shapes how I approach the conversation that we're going to have today. So let's do it. Let's start talking about what research really is. All right, so there's a lot of things on this screen, but we're going to simplify it. When we hear about new treatment, it can feel like progress should happen quickly. In reality, treatments are studied step by step over many years. And here's the thing, that's intentional. Each stage exists to answer a specific question, and together they form a system designed to protect patients and produce reliable answers. So at the earliest stage, preclinical, research happens outside of patients. It's in labs, disease models. It's kind of what you think about when you think about clinical research. You think about people in lab coats with beakers and animals in cages behind them. That's where researchers ask whether or not an idea makes biological sense, whether there's a signal that it could help, and whether there's safety concerns that we need to think about. Thousands of ideas may be explored at this stage. But here's the thing, only 5 to 10% will move forward. That means most potential treatments stop here before anyone with CMT is exposed. This stage can take anywhere between 1 and 6 years. So here's the thing, if a treatment does pass preclinical testing, it's going to move into something called phase 1. And this is the first time it's given to humans. These are small studies. They're focused on safety, how the body handles the treatment, and what dose appears to be reasonable. Out of 10 treatments that enter phase 1, roughly 6, 7 will continue on. And here's the thing, phase 1 is not designed to show benefit. Any improvement seen at this stage, it's considered exploratory. Now, phase 2 is where the rubber meets the road. This is probably the most challenging stage for us as drug developers. These studies involve more people, focus on identifying the right dose and looking for early signs of benefit while all continuing to monitor safety. And this is where we see the largest drop off. Of the 6 or 7 treatments that enter phase 2, only about 1 or 2 or 3 will move forward. Many treatments are safe, right? They're safe, but they're not effective, or they're not effective enough, or they're not effective in the right way. And this phase is often where expectations meet data. So if we move past phase 2, we're going to go into phase 3. And these are large, they're rigorous trials, and they confirm whether a treatment truly works and whether benefits outweigh risks. Usually at this point, only 2 or 3 treatments reach this phase. And of those, only 1 or 2 may succeed. And so after a successful phase 3 trial, you have to put the trial through regulatory review. This is where we review the data. And this process can take anywhere from 6 to 18 months. And even after approval, we continue learning. We go to something called phase 4 studies. This is long-term monitoring. This is what the drug is like in real world. So here's the big picture takeaway. From early lab research to an approved treatment, it typically takes 10 to 15 years from molecule to medicine. And about 10 treatments that enter into human trials, only 1 will become an approved medicine. And that's not because research is broken. It's because patient safety comes first when you're doing drug development. I get it. It's slow. But in research, slow means careful. So next slide. So what's the difference, right? Another common question that we get a lot is the different types of studies that people hear about. So here I'm going to talk about two: observational and interventional trials. They're both research. They're both very important, and they both serve very different purposes. In observational studies, researchers are not testing a treatment. They're learning by observing. And this relates to CMT about how it affects people over time. Participants continue their usual medical care. They get their standard of care, and the study doesn't change any treatment. Information may be collected, and this is done through questionnaires, exams, functional assessments like the 6-minute walk test, and sometimes genetic and lab data. These studies help researchers understand disease progression. It helps us identify meaningful outcomes. And here's the most important bit: it helps us design better trials. An opportunity to participate in an observational trial helps us shape how we build interventional trials. Now, interventional trials, they're different. In an interventional trial, researchers are testing a specific treatment. For example, a medication or a gene therapy. These studies follow a very structured protocol that outlines exactly what happens, when it happens, and how safety is monitored. Participants in these trials may receive study treatment, or they may be compared to another treatment and another approach. These trials involve more visits, they involve more testing, and they're very closely monitored. And again, I go back to this is very intentional. As researchers, we closely track safety, side effects, and how your body responds to it, often with more oversight than you would receive outside of a study. So participation adds a research team focused on learning and safety alongside your regular medical care. And here's the thing: both studies are important and help improve future care for people living with CMT. Next slide, thank you. All right, so I'm really excited to talk about this slide. This is a slide that I've used personally with my family. And here's how I started it off. There isn't a single moment where someone suddenly joins a clinical trial. It's a sequence of steps. I used this chart when my husband wanted to be part of a phase 3 study because I really wanted him to understand what he was signing up for. And so I'm going to walk you through the same way that I walked this through with him. And I hope it helps answer some questions. So it starts off with a diagnosis. Everything starts with a diagnosis, whether it's recent or it's a long-standing diagnosis. So for my husband, he didn't have a diagnosis. We knew he had CMT1A because my daughter had been diagnosed at age 4 and my father-in-law had been diagnosed, but my husband had never gone through genetic testing. So before he even started his clinical research journey, he went and he got diagnosed formally. What that does is it helps us identify the CMT subtype and whether there are certain research opportunities that might be relevant for you. Now, the next part of this is the referral. And I really hate this word because it doesn't really translate to what's actually happening. But a referral just means that it's connecting you from you to someone in the study team to learn more. And this referral can come from your neurologist or a specialty center or an advocacy group or personal research. When we were ready to do our journey into clinical research for CMT, we found the trial that my husband was on through clinicaltrials.gov. We went, we did our personal research, we found it, and then we called the clinical site that was participating in that trial, which led to our next step, pre-screening. And so pre-screening usually takes place on the phone. It might be an online conversation. Sometimes we do it in person. And this is where the study team reviews eligibility. So for my husband, they asked him about his age, his subtype, his medical history. And this step helped him, my husband, and the clinical site determine whether it made sense to even continue the conversation. Pre-screening is a very important part of the process. It helps us begin that journey. But the next step is the most important part of the process, and that is informed consent. Participants receive detailed information during the informed consent process. We talk about the study, including its purpose, what participation involves, potential risks, alternatives. And here's the thing, this is the time to ask questions and consider all the information carefully. When my husband was ready to do this, we brought the consent home. We read it as a family. We digested it all. We figured out whether we were going to be able to fit this into our lives. And we went back and we asked questions. How much time is this going to be involved with this? What happens if I want to stop? Will I continue seeing my regular doctor? And what do I do if I have concerns? Who do I talk to? Nothing proceeds without informed consent. So if you move forward, you choose to want to go through the study, the next phase is going to be the screening phase. Now, screening phase can be anywhere between a day, it could be a month, and screening visits can be long, they can be short. For my husband, when he did it, his screening visit was a day. It was a full day, and it's where they confirmed his eligibility. They went through safety labs, they did exams and assessments, and it was during that time they determined whether or not he met the inclusion criteria within the protocol or he didn't, and he would be excluded. Now, there's nothing wrong with being excluded. It just means that it wasn't the right fit for you to be part of that study. For my husband, he was included in the phase 3 study, and then he was randomized into a study group. And this will all be explained through the informed consent process. Now, after he was randomized, he started his full participation in the trial. And so it included scheduled visits, ongoing safety monitoring, regular communication with the study team. And lastly, before we move on to the next slide, here's one important thing. At any point throughout this process, you can leave the trial. So for my husband, he went through the entirety of the trial and he got onto the long-term extension that was part of his phase 3 trial. And he stayed on there for about 7 months, but the time commitment got to be too much. And at that point, at 7 months after he was on the long-term extension, he decided that it was time for him to leave the trial, and it was completely fine. No, all right, so I think next slide. Perfect, thank you. So I've talked about clinical trials, but what a very practical question. Where do I look for studies? So start with your neurologist or your care team. They can help identify studies that may be relevant or help interpret it and help interpret the details. Patient advocacy groups like the CMT Research Foundation, they help curate CMT-specific opportunities and provide plain language guidance. Large hospitals, specialty centers, and university medical centers frequently run studies that they might not always be widely advertised, but they're available. And then trial referral services exist as well. So we've all seen those. You're on Facebook, you're scrolling through, you pop up a reel that says, hey, do you have migraines? Click here. So those opportunities, those are referral services, and they will match you to where, potential opportunities. There are multiple sources. So your care team, your advocacy groups, public registries, and that all help to build a clear picture of what's available. So if you go to the next slide, this is what I was talking about before, the clintrials.gov. This is how we found our trial that my husband participated in. Now, I'm not going to go through all of this. I think we know how to go in and type in CMT, but I will say this, that it is a very easy website to work through if you follow the specific prompts. But you have to be very careful about what you put in. Put in Charcot-Marie-Tooth, put in CMT, put in recruiting or not recruiting yet, zip codes, put in, the age you're looking for, and it will curate a list for you. And I think the best thing is that I think it's available as a link on the CMTRF website. So you can click right on it and it'll take you to clintrials.gov. So let's wrap up and bring it all together. I want to take a step back and look at the bigger picture. CMT is personal. It affects real people like me, like you, real families, and our real lives. Research is how progress happens, how we learn more about diseases, improve care, and work towards treatments that make a meaningful difference. Understanding how that research works helps set expectations, and it reduces your uncertainty. When my husband walked into that clinical research trial site, he knew what he was up for. It made him more informed, and it allowed him to ask the questions that he needed and to engage with his care from a position of knowledge. Clinical research in CMT, it's evolving. The field is more targeted, it's more collaborative, and it's more patient informed than ever before. We're seeing it. When my daughter was diagnosed 13 years ago, no one knew what CMT was. There was barely anything out there. There was nothing that we could search. And now there are famous people that are out there with CMT. People actually know what we're talking about sometimes. It's there. It's becoming more mainstream. It's where it needs to be. The science is getting better and making the right decisions earlier. And this is what matters. So that's it. My slides. Thank you.
Thank you, Jenneen, for that fantastic overview. It was very helpful. I learn every time. I learn something every time that you talk about it, which I appreciate. Even having a background in drug discovery, I think it's great to understand the nuances, also to think about the patient perspective, and really keep that top of mind, right? Like, what are the practicalities of this? And also from the, drug development perspective, right? How do we keep the patient foremost and their experience foremost? Because
Yes.
ultimately that's what matters. I mean, you hope to design it in a way that people don't want to drop out because participating is something that is, meaningful and in line with what they're able to participate in. So
Yeah, the criticality of removing barriers to participation is probably the most important thing when we're designing programs.
absolutely. Well, so one thing that I'd love to walk before we take questions, I'd love to walk everybody through our a section of our website where we stay up to date on clinical trials for CMT. A few years ago, this would have been pretty empty, and it's really exciting to see where we are today. So we've broken it apart. We have a section that, first of all, describes, in very simplified terms what Jenneen just talked to you about. So we'll reference Jenneen's video from here on out. But we have at the very top, we have active trials that are recruiting patients. So you'll see that we have two, one by Novartis Pharmaceuticals, and the other is by Ncell. Novartis is a pretty big name in pharma. A lot of people have heard about them. They're currently in a phase 1 trial advancing their molecule called EKD-060. This is a single dose of the molecule. It's an IV infusion. And now, as people have learned from Jenneen, phase 1. So this is really looking at safety and focused on that. Currently, they have two clinical trial sites active. They're recruiting, both of those sites are out of Canada, and they aim to recruit 28 patients with CMT1A. The next one that you'll see here in the active trials recruiting patients comes from a company out of South Korea called NCell. So this is an injectable stem cell therapy that's thought to remyelinate nerves and help to rebuild muscles. Right now, they're in a phase 2. They've completed a phase 1B. Believe they're moving into a phase 2. They're recruiting additional patients with both CMT type 1A and type 1E. And this is a trial that's based out of South Korea. Below that, we have a section on trials, and these are ones where recruiting is complete. So there's less for you as a patient to do in terms of, can I get involved somehow? This is more like, keep an eye on these guys. Let's see what data is forthcoming. Often once recruiting is complete, there can be several months of follow-up and then time to process and digest and understand the data to then be able to talk about it. So among this, there's applied therapeutics. This is for a type of CMT that's specifically caused by mutations in a gene named SORD. So it's sometimes called SORD CMT. It's a type 4. Recruiting is complete. They've analyzed the data. They're working on what are their next steps. NMD Pharma also recently completed enrollment of their trial. They enrolled 81 patients total across types 1 and type 2. This was for an oral drug that's taken twice daily. If you attended our convention earlier in September, we heard from a patient who talked about her experience on that trial, and it's really neat to hear from the patient perspective what that was like. So NMD Pharma is going to be soon. They're also in the process of analyzing, digesting their data, and keep an eye on next steps from them. It's another trial, Nationwide Children's Hospital. So this is driven by Dr. Megan Waldrop, who is a clinical scientist. This is an investigator-initiated trial to look at intrathecal or IT injection. You might hear of it. That's where you inject directly into the space around the spinal cord. This is with a harmless virus that carries a copy of a gene that causes CMT type 2S. So this is in a phase 1/2, and again, it's at Columbus, Ohio. If you have interest in CMT2S, you can click to learn more, study to get in touch with Dr. Waldrop. Lastly, in the active but no recruiting is Augustine Therapeutics. And so they've been investigating a small molecule that is taken orally that is meant to support nerve health and reduce degeneration that's caused by CMT. They're in a phase 1 study. The recruiting is complete, and we'll be awaiting more data from Augustine as well. In development, there's 2 trials of note. One is by Elpida Therapeutics. This is another gene therapy that uses a harmless virus to deliver a good copy of the gene that's mutated in patients with CMT type 4J. This is caused by mutations of the FIG4 gene. Similar to the trial that I described that Dr. Waldrop is conducting, this is a trial that will inject that harmless virus with a single injection one time into that space around your spinal cord, known as an IT injection. Right now, they're in the planning stages, and we'll await more data, and once we have it, we'll update the site. Similarly, Actio Biosciences, who's exploring a small molecule to be orally administered. This is for CMT type 2C. They've completed some natural history studies and are planning their clinical intervention studies, first to evaluate in a phase 1, and then hopefully also in a phase 2 to look for efficacy. So as Jenneen mentioned, there's both interventional trials, and that's what I just covered, but then there's also observational studies, sometimes called natural history studies. And so we have a curated list here of many ways to get involved in a natural history study. And as Jenneen said, these studies are very important for understanding how a disease progresses in a patient so that we have a better idea as clinical trials are designed, how best to design those clinical trials to be able to capture optimal outcome. And so, again, if you have any questions, feel free to reach out to us. We hope that this site is valuable, and we'll continue to keep it up to date. So now I'd like to turn it over to Susan, who's going to moderate our Q&A session with Jenneen.
Thanks, Meg. Thanks, Jenneen, so much. So just so everyone here can see, we have about 25 minutes for questions. We really wanted to be very condensed with our presentation because we know that this is one of the biggest topics that people have questions about. So pop them into the chat under the Q&A and we'll be answering them. And so I want to start with a question that came in. And one of the things that's really cool, Jenneen and Meg, that you showed us is that CMT is really a global issue and that it's really a global effort. Meg, you were talking about trials in Korea and in Canada. We saw some observational trials happening in Italy and a lot happening in the United States. If we see a trial that is taking place in a country where we don't live, is there anything that we as patients can do to bring that trial closer to us? Or is there anything that we as patients can do to go overseas to participate in these trials? I mean, talk to me about this process.
So I think I can talk a little bit from a drug development perspective, right? I think when we're designing trials, we're very bespoke on what locations that we're picking. And in that case, though, there is sometimes an opportunity, but it's really up to the drug developer whether they're willing to go down this road around something called cross-border enrollment. And what this means is that you can actually travel to that country to get that drug. Alternatively, you can petition the sponsor or the biotech, the biopharma to potentially open up a study within your country. So there's different routes. It's hard to do, but it is potentially possible. The other thing is that if you're early in the clinical development pathway, say you're in phase 1 and it's maybe going to phase 2, well, if it goes to phase 3, that's going to be a much larger program. So there is going to be an expansion at that point. Typically, you'll see if it's in 1 or 2 countries, maybe it's going to span to 5 or 6 or 7 countries. So there'll be more opportunities at that point. There's definitely a mechanism, but it is a challenge to do that.
And forgive me if you were answering this question in your answer, just because it popped up and I wanted to make sure I captured it on the same thing. Um, if a trial is not currently happening in a country or in the United States, even let's say, um, we would assume it's because the regulators or the pharma company wasn't prepared to bring it to the United States at that time. But we hope that it can and will come. Can you talk just for a sec about that?
Yeah, so it goes into a little bit about, that strategic site and country footprint, right? So it might not be where it's feasible to do a study within the United States. It could be an expense reason, it could be the regulators. But from that perspective, again, get your voices out there. If this is something that you would want, make sure that the drug developers are hearing that, because there's always opportunity to expand, right? So I think just be active in the community, talk to the individuals, but it does have a lot to do with how we set up the countryside footprints, the regulators, all the different mechanisms that go into developing a clinical trial.
Thanks. Thanks for expanding on that answer, Jenneen. So one of the questions that we have, and Jenneen, I'll start with you, and then Meg, I'll turn it over to you. But we're getting a lot of questions in the chat about like, well, what about my type? I'm not seeing my type referenced on this. And are there some trials happening that are for many different types? And, um, it's not clear, but they might be for many different types. There are some trials that are very type specific, like the 1A and the SWORD trials that we heard about. And then what happens if I don't have a genetic diagnosis? If even I've gone through the whole process and my genetics aren't clear? Can you talk a little bit about what kind of hope there is for me for my type, even if I don't see it?
So we're definitely looking at this from a different lens, right? There are so many subtypes with CMT. There are trials that specifically they're called all comers, and we've seen some companies are now thinking, well, should I do an all comers study? That would potentially allow people who might not have a clear diagnosis in. It could allow many different subtypes. The other avenue that I would really encourage you to go down, there might not be an interventional study that's set right now for your subtype, but observational studies, natural history studies, we need that data as well. Because if there is an opportunity for us to be able to study your subtype, we're going to need some reference natural history data that comes from that. So I know that there are a lot of natural history studies out there. If you can participate in one, that would be fantastic. But I think the future is brighter when it comes to the different subtypes. I think we're going to see more all-comer studies. I think, again, we're seeing a blossoming of the CMT world. So I have hope for it.
Great, thank you. And Meg, will you speak specifically about some of the trials that are for all types and some that are for specific and maybe why?
Sure, yeah. So when we see a trial that's for, I'll start with the specific types. So frequently when we see a trial that's for a specific type, that type of therapy is specifically addressing the underlying genetic mechanism. So one of the things that we know about CMT and that we know a lot about with regard to CMT is the underlying genetics. And so when trials are recruiting for a very specific subtype, that's typically because it's doing something to affect that genetic change. So for instance, if it's a trial for type 2S specifically, that means that it's addressing the 2S gene. If it's specific for 1A, then it's typically going to do something to reduce PMP22 levels. And reducing PMP22 levels is something that would be unlikely to affect or to have benefit for another type 1 patient who doesn't have a mutation of PMP22, but also has a demyelinating form. You know, for instance, type 1B is caused by a mutation of a gene called MPZ. And so for something to address the gene that causes or the overexpression caused by type 1A may not benefit a patient who has type 1B. For some of the therapeutics, though, are to support neuronal health generally. So I mentioned the NMD trial. I mentioned the Augustine Therapeutics trial. I'll just stop at those two right there. They're both drugs that are aimed to support neuronal health overall. And so whether you have demyelinating or axonal, those mechanisms can help that nerve to function better. And so there, the specific subtype is going to be less important. And you might see more that they're recruiting people who have type 1 or type 2. Sometimes they may still require a clinical diagnosis. And the reason for that could be to help make sense of the data afterward. You know, so for instance, if they don't see a uniform response across all patients, it would be very helpful for investigators to have some information around the genetics of the patients enrolled so that they could stratify that data and see if, one particular subtype or genetic mutation had a better response than another. Hopefully that helps to clear up that or answer that question.
Yeah, thank you. And for those of you who are listening, if it didn't answer your question, just type in more questions. We've got a lot here that are open, but type them in because we still have some time. Jenneen, one of the questions that has come up, and we've heard this a number of times, is it possible for me to be too old to participate in a trial? Or could a recent surgery disrupt my eligibility of being in a trial? And
Mhm.
so, can you talk a little bit about how companies make their eligibility decisions and why they're so important and how people who might be on the outside of those eligibility decisions can think about
Mhm.
participation?
Yeah. So everything starts and begins with the inclusion exclusion criteria. And they're so vitally important because within the power of a protocol, we are testing to ensure that we can see a sustainable change, right? That's what we're looking for. We're looking for a modifier, right? And so there might be instances where that exclusionary factor, your age, you might be plus, 100. You might have had a recent surgery that would impact maybe your pain scores when you're doing a daily pain test, right? These can all impact the trial, how it showcases the risk benefit, right? It impacts the results, then we get skewed data. So they have to be very prescriptive at the beginning. And it's not, again, it's not to say that you won't always be able to participate. It's just in that particular trial, it's probably not going to be the best fit. We want to make sure that we can get the data across the finish line with the regulators, and we have to have the best data possible to move forward.
Yeah, just a little personal anecdote there. I had hoped to participate in a trial this summer, and I had gone through the initial screening, and I was deemed a good candidate. And I went out for a walk and tripped over my shoelaces and shattered my kneecap and required immediate surgery. So therefore, I became immediately ineligible for that trial. And the way that the pharma company described it to me was, Susan, we want to be able to assess your activity now as your baseline. And then if your activity improves, we want it to be because of the drug. Well, right now, you're completely immobilized with a broken kneecap. And in 12 weeks, you're going to be moving a lot more because your knee will have healed. And that will be the natural process, not the drug that's making you feel better. So you're going to skew our data. And while we'd love to have you, thank you, but no thank you. It was a heartbreaking moment. Actually, as I was laying there on the ground, grabbing my knee, knowing the surgery was coming for me, I thought, that's it. No trial for me this summer. So I had to be patient and wait. But I do one thing I did hear from this company in particular is that once my activity levels were kind of leveled out and I was back to whatever my new normal was, after you have a major surgery like that, maybe you get back to pre-surgical levels and maybe you don't. So once you have those new normal levels and you're back moving again in your new normal way,
Mm-hmm.
that would be a good time to see if you're eligible. Does that
Absolutely.
track with what you know?
Yeah, that's absolutely correct. So when you can get back to your physicality, we can start measuring things. You're not going to see the data with your results. Absolutely. Go back, have that conversation again. We call that rescreening. If you've screened, and you screen fail, then we can rescreen you. If you don't even get to that point, well then, this is now your first rodeo doing this, but we absolutely want patients to come back. If you're not eligible this time, you might be eligible the next time.
Yeah, thank you. Meg, one of the questions that we hear quite a bit, and one of the positions we have at the Research Foundation, is that a gene therapy for one type of CMT, while it's very specific for that one type, we will have key learnings that can unlock gene therapy development and trials for many different types, and especially in the ultra rare community. We've got a couple questions here talking about the ultra rare types of CMT and how gene therapy really does feel like the best option for them. But obviously, the only gene therapy trials that are going on right now are for 2S and about to start for 4J. So, can you talk about, especially people in the ultra rare community, but with all types of CMT, how they can monitor this LPTA trial and this 2S trial and what that means to them? And are there ways that these trials can speed up the process for them?
Yeah, absolutely. It's a great question and something that in the development community, especially development for rare disease, we think a lot about, right? How can we build something that can also help others, and help accelerate timelines? So in terms of monitoring the progress on those trials, look for public presentations. I'm guessing that we haven't set our convention for next year. We have the date, but we haven't set the complete programming. Hopefully we'll have some updates from investigators at that time. For the Elpida trial, we expect that it'll be a little while before we can say a whole lot about it. And some of that has to do with the way that the trial is designed. Typically with trials like this, you start with a very conservative approach where you will dose a patient, there'll be a waiting period. You dose the next patient, there's going to be a waiting period. And so it's going to feel slow, but it'll tell us a lot of important data that will help to accelerate programs in the future. So there's important questions around safety. These are moved forward because we do believe it is in fact safe. Right? So I'm just going to start there, right? Is that it wouldn't move forward with an adverse safety profile. But in order to gain more knowledge about how this behaves in a person, right, and how well that virus delivered to that interthecal space, the IT space, how well that distributes throughout the body, specifically to the peripheral nervous system, if patients respond, these will all be learnings that we can then say, aha, we know which harmless virus we can use to deliver a therapeutic cargo. And we know whether or not, and we suspect the answer is yes, because that's how it's designed, right? That if you deliver into that interthecal space, that you're going to get what's called the biodistribution. So where this drug goes in the body, you're going to get the biodistribution that you need. The other thing is that it'll help us to understand better how to study what Jenneen went over, endpoints. Right. So every trial, whether it's a gene therapy or otherwise, seeing how patients respond and how we measure that response and how responsive that particular measurement is, right, to the drug, to the therapy, is very important for informing the next trial and making that that trial more accurate, and to give us the best and most interpretable results.
Thank
Does
you.
that answer the question?
It does for me, but again, if it didn't for you and you're an audience member, type more in the Q&A and we'll get to it. Okay, so on to the next question. Another question that is coming up quite a bit in different formats here is, my type is unknown. Where can I get genetic testing? How can I get that? Why is that important? What happens if it comes back and it says, we really don't know what type you have? Can you talk a little bit about the diagnostic process and maybe some of the people who we're affiliated with who are geneticists? I know you are, but you're not doing genetic testing. But can you talk about some of the resources that we have at the Research Foundation and maybe how patients can go about finding genetic testing?
Yeah, absolutely. So it starts with a conversation with your clinician, right? So, and your clinician can order a genetic test. There's commercially available genetic tests, and they have what are called gene panels. What that is it's a list of genes that are known to cause CMT. And so, what's typically the very first step, typically with genetic testing, is to screen patients for PMP, for duplications of PMP22. That's the cause of CMT1A. And about 50% of patients, sometimes 60, depending on the number you see cited, will have that mutation, which is why upfront you screen for CMT type 1A. If that's not the case and you go for expanded genetic testing, that involves the entire panel of that captures the majority of CMT related genes. And so then they'll take either what's called a buccal swab. So that's when they swab the inside of your cheek and take some cells off of the inside of your cheek. Looks like a little mascara brush. And or you can draw blood, or sometimes there's spit kits, and what that spit kit is because also you're taking some cells that slough off in the inside of your cheek. And then that's sequenced. And then if you have a mutation of a known gene causing CMT, or that is known to cause CMT, you'll get a report. There are reports on that particular, you have this mutation is in this gene, and that's causes, CMT type, I'll say 1B, right? Sometimes your report will give you something that says that you have a variant of unknown significance. So it might say we identified a genetic change in the gene that causes CMT type 1B, but we can't say with certainty that this particular genetic change will absolutely be the cause of your disease. That's where you get the variant of unknown significance. And it's a very cautious approach taken by geneticists because not every mutation is going to be disease causing. Our bodies just pick up mutations. Well, throughout evolution and the development process, mutations will accumulate in the human genome, and not all of them can cause a problem. And the more evidence that you get behind a particular mutation, the more certainty that you can have that that mutation is causing CMT. And so that's where increased genetic testing, increased awareness around it really helps the entire community. There's databases you can scroll through, and what's kind of neat is in these records, it's called ClinVar. You can see where patients have been identified to have a particular mutation. It's completely anonymized. Nobody knows who it is, but you can follow the identification history of a particular mutation as you go to building a body of evidence that, oh, actually 10 people who were suspected to have CMT have reported to have this mutation. Now we're not going to say that this is a variant of unknown significance. This is likely to be what's called a pathogenic or disease-causing variant. So talk to your doctor, check out one of the commercially available gene panels. And then also there's the INC, which is the Inherited Neuropathies Consortium. And you can visit their website for more information as well to find a center of excellence where they can point you in the direction of genetic testing.
Thank you. Thank you. We have on the line actually one of the world's best geneticists from Australia, Marina Kennerson. She's on this webinar today. Thanks, Marina, for joining. And she's also on our scientific advisory board. So if you're in that side of the world, you can reach out to Marina. You can reach out to us, and we'll get you connected to work with your doctor to get you connected to her. Also, Dr. Wolfgang Pernice is at Columbia, and he's affiliated with the Research Foundation. He's a geneticist in Columbia University in New York City. Stefan Zuckner at University of Miami. These are all resources that we can help get you connected to when it comes to genetic testing. But working with your primary doc is the best way to start. Um, we only have 4 minutes left for this formal part of the webinar, and then we're going to open it up into a less formal segment. And we're going to stay here until all of your questions are answered. Just so everyone here knows, I still have 12 questions in the Q&A thing. I want to close with one question. But for those of you who ask very specific questions about your types, that's where we can get into maybe a little bit of a conversation in the less formal side. I know that there are questions about HNPP, and there are questions about how do I know the difference between type 1 and type 2, and some of those questions that are a little bit more specific. So we'll get to those later. We're not going to forget you, I promise. Um, there is also a question about how patients can help advocate with the FDA. Thank you for the person who asked that question, because the answer is connect with us, because this is something that the Research Foundation is doing on a regular basis. So if you are interested in being an advocate to the FDA to help them understand CMT, please reach out to us and we will help get you activated. So my last question is from one of our participants in Australia who we've met several times. And she is asking, given the advancements in increasing knowledge about CMT and giving the technological advancements like AI and the range of trials mentioned, how are we going to be able to reduce the timelines? Because these timelines that Jenneen gave us are pretty daunting. And as a 54-year-old woman, oh, I'd like to get this in my body sooner rather than later. So how can these technologies and these learnings be leveraged to reduce this time for parents who are watching their children degenerate, or people who are watching their own bodies? How can we have some hope in this timeline with the current technologies?
So I think I can start here. It's still really, really early, right? These are newer technologies, but you're right, the hope is there with AI technologies, and how do you optimize clinical research so you can accelerate, right? We're at the beginning. I will tell you that it is the thing that we're seeing at all the conferences. Everybody is talking about AI and how we can do this. I I don't know the answer to the how, because I think we're still building the how right now. But I know there is definitely a when very near in our future. We're building the bus while we're driving it right now. I'm going to a conference called Scope next week. It is littered with AI technology. It really is what I think is going to be the promise of a much more accelerated drug pathway.
Thank you. Meg, what do you think?
Jenneen did a great job of answering that. I agree with a lot of what Jenneen said. I think that, in terms of how we accelerate trials and the role of AI in that, one thing about AI and machine learning, so particularly in science and diagnostics, and also, using it as a clinical endpoint or to help support clinical endpoints, is it takes a lot of learning. Actually, not to sound redundant, right? But it is in fact a machine learning. And so you have to give it a lot of presentations of what you're looking for and what, a healthy looks like and what a CMT walk or what, like a CMT muscle. You have to give it that information and say, look for this. This is what you learned from. And so some of that I think will ultimately accelerate or, and help to support development, but it's going to take a little bit of time to get there, right? Because you have to feed it that body of information in order to train these models to know what to do with it, and then also validate the accuracy of it.
Okay.
And that validation of the accuracy of it is critically important because you want to be able to trust whatever results are coming out. So I think there is, in fact, an important role. I think it will accelerate, but it's not like ChatGPT, right? It's not there yet because we need it to be, we need to train these AI doctors.
All right. Well, everyone, it's 8 o'clock. And we like to keep to a pretty tight schedule, respecting all of your time. So, you are welcome to leave. You've heard the meat of it. And for being here, I just want to say thank you so much for joining us tonight. Thank you to Jenneen and thank you to Meg for being here.